Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing University of Hyderabad PhD Entrance Examination Biotechnology question paper conducted in the year 2022. I will be discussing only the biology part. And if you are new to my channel, please do like, share and subscribe. To the first question. Following structure exhibit dipole moment. So it is alpha helix that exhibit dipole moment because a helix has an overall dipole moment due to aggregate effect of the individual microdipoles from the carbonyl groups of the peptide bond pointing along the helix axis. So the answer here is option A. Then, if a protein shows 4 conformations C1, C2, C3, C4 with potential energy of minus 40, minus 50, minus 70, minus 30 kilojoules, which is the stable conformation? So, minimum potential energy, maximum stability. So, minimum potential energy is for C3, that is minus 70 kilojoules. So, answer here is option C. Then when protein is in a given sample are precipitated with ammonium sulfate at different saturation concentration, which percent saturation a hydrophobic protein could be detected? So at 80% saturation, hydrophobic protein could be detected. So answer here is option D, that is 80%. Then next one, which of the following microscopes are used to visualize the protein fused to an appropriate reporter in a living cell? So it is fluorescent microscope. Answer is option B. The next one, for glycoproteins, most commonly used probe is lectins. Answer is, because they are glycoproteins, so lectins are used as probes. Answer is option C. The separation technique of charged molecules under the influence of electric current is called electrophoresis. Answer is option D. The next one, the identity of an element is determined by its number of protons. Answer is option A. Then next one. During the separation of proteins using SDS page, the following two dyes are used. So which are the two dyes? So one is bromophenol blue and then is Kumasi brilliant blue. So 2 and 3 are used. Option C is the right answer. The next one. To construct a recombinant plasmid, the plasmid and the DNA to be inserted are digested with the same restriction enzyme and added to the ligation reaction mix. Which of the following enzymes can be used to prevent self-ligation? So alkaline phosphatase is usually used to prevent self-ligation. So answer is option. Then next one. What is the final pH of a solution made by mixing 40 ml of 0.3 molar HCl and 20 ml of 0.2 molar NaOH? I have given the calculation here in detail. You can go through that. The answer will be 1. Answer is option A. Then next one. Which of the following compound is used as an internal reference in NMR spectroscopy? So it is tetramethyl silane. Answer is option D. The next one. Which of the following parasites belong to phylum Epicomplexa? So it is Plasmodium falciparum. Answer is option B. The next one. Choose the right answer. B DNA is left-handed. It is not left-handed, it is right-handed. So A is wrong. Then B DNA is again they have written left-handed, it is right-handed. So that is also wrong. B DNA is formed when relative humidity is 92%. That is correct. A DNA is right-handed DNA helix with 10 base pair per turn of the helix. It is 11 base pair per turn of the helix. So D is also not correct. So the answer here is option C. Then next one. The ionic strength of 0.2 molar K2SO4 is dash mole per liter. So for that also I have done the calculation over here using the formula half CiZi the whole square, where Ci is the concentration of the ith ion and Zi is the charge of the ith ion. So you can refer to that calculation. I have written in detail. Then next question. In the given CD spectra, which line indicates beta sheet? So it is the line 2 that indicates beta sheet. Answer is option B. If two organisms show region of conserved synteny. So what is synteny? It describes physical co-localization of genetic loci on the same chromosome within an individual or species. It means that orthologous genes examined in the two species are, so they are present in the same localization order on a chromosome. Answer is option A. Then next one. Dot matrix can be efficiently utilized. So it can be utilized to graphically visualize regions of similarity between two sequences. Answer is option B. Then next one. Which of the following sequencing technique is used to detect methylation levels in DNA? So it is bisulfite sequencing. Answer is option. Then next one. Cellular organelles involved in Ca2 positive level in the cell. They are mitochondrion and endoplasmic reticulum. Answer is option D. The expression of transgene in a target tissue is identified by. So it is identified by the reporter gene. Either GUS or GFP is used. So answer here is option B. Then Swissport, it is a database which deals with proteins. Answer is option D. Then Riesc iron sulfur protein is located in one of the following complexes of mitochondrial electron transport chain. So it is located in complex 3. Answer is option C. Then data retrieval tool of NCBI, that is Entris. It is data retrieval tool. Then, which of the following amino acid has pi value more than 10? Of course, it is arginine out of the given amino acids. Answer is option D. The melting temperature of DNA in distilled water is, it is room temperature. Answer is option A. Then next one. Which of the following amino acids are non-essential? So they are alanine, then comes aspartate, and then comes glutamate. These are non-essential amino acids. Answer is 1, 3 and 4. Option B. Then part B. First question. The virus that integrates into the host genome but does not require for its replication. Hepatitis B. Answer is option A. Then next one. Non-covalently linked DNA intermediates formed during homologous pairing are known as. So they are known as joint molecules. Answer is option A. Next one. In an enzymatic reaction, when the activity of inhibitor was monitored at different concentrations and 1 by V and 1 by concentration of S was plotted at different concentration, the graph shows an increase in both the slope. Slope is your Vmax and slope of the graph and intercept on the y-axis, that is Vmax is increasing with increasing concentrations. What would be the affinity of I? So I think this is a case of mixed inhibition. So it will be enzyme only or enzyme substrate complex only. So answer is option C. Then next one. Ligand-based drug design involved regression of biological activity with molecular descriptors. Answer is option D. Energy minimization method for optimization of a molecule using both search direction and decrease in energy is conjugated descent. Answer is option A. Which of the following cellular event is not directly associated with mitochondria? So it is apoptosis, ATP synthesis, cell cycle regulation, protein degradation. That is not directly associated with mitochondria. Answer is option D. The next one. Which of these compounds generate a feeling of well-being and inhibit sense of pain? So it is endorphins. Answer is option D. So coming to the next question. The vaccine prepared through recombinant technology is, so it is third generation vaccine. Answer is option C. Then which of the following statement is true? Alleles cannot admix, so hybrid alleles do not usually occur in a population. That is not true. A particular gene can have only two alleles. No, there are multiple alleles also. Pseudogenes occur frequently in viral genomes. That is also not true. A single trait or a phenotype can be affected by many different genes. Yes, that is correct. Answer is option D. D is true. Then next one. Find out the correct order of steps involved in the development of a bacterial phylogeny or ancestry. Isolation in pure culture, reduction of a distance matrix, construction of a network or tree, DNA extraction, analysis of DNA polymorphism through fragment analysis or sequencing, validation of tree topology and inference of phylogeny. So the answer first one will be isolation in pure culture. Then comes DNA extraction. Then comes DNA extraction. Then analysis of DNA polymorphism through fragment analysis or sequencing. Then comes deduction of distance matrix. That is your fourth one. Then come construction of a tree or network. That is your fifth one. And validation of the tree topology and inference of phylogeny, that is your sixth one. So it will be 1, 4, 5, 2, 3 and 6. Answer is option D. Then consider the following statements. Which of the following are correct? 4 carbon acids are required for fixation of carbon dioxide in bundle sheath cells. Yes. And first one is correct. Light plays a role in activating C4 cycle enzymes NAD, MDH, PEPCase and PPDK. That is not correct. Light activation of NAD, MDH involves the ferredoxin thioredoxin system. No. And 4. Light activation of PEPcase and PPDK is regulated by allosteric effectors and protein phosphorylation. So 1 and 4 are correct. So answer is option C. Then next one. Fluorescence is emitted by one of the following compounds during Roche analysis in a living cell. So it is DCF or dichlorofluorescein. Answer is option B. Then next one. Alternate oxidase particles in mitochondrial electron transport chain. Identify the correct statements with respect to AOX. Alternate oxidase. AOX is found in plants, algae, fungi and protozoan. That is correct. AOX transfers electrons from ubiquinol to oxygen bypassing cytochrome C oxidase. That is also correct. AOX transfers electrons from ubiquinol to oxygen. That is not. But bypassing cytochrome C oxidase has also to be there. So 3 is not correct. 2 is more correct. AOX is encoded by mitochondrial genome. It is nuclear genome that encodes AOX. So the answer here is option A, that is 1 and 2. And in third statement, it is not ubiquinone, it is ubiquinol. Okay. Then coming to the next statement. Identify the correct statements from the following. Sucrose and PI are inhibitors of sucrose phosphate synthase. That is correct. Fructose 6-phosphate and PI are activators of fructose 2,6-biphosphatase. Fructose 6-phosphate and PI are activators of fructose 6-phosphate 2 kinase. That is correct. Fructose 2,6-biphosphate and PI are inhibitors of fructose 1,6-biphosphatase. That is also not correct. So 1 and 3 are correct. Option B is the right answer. Then which of the following is not an approach for local alignment? Smith, Waterman, K-tuple, Bird method, Needleman, Wunsch is for global alignment. So option D is the answer. The metabolic database is KEGG. Option A. Which is the correct group of sentences for neglected tropical diseases? They are called neglected because they generally affect the world's poor and historically have not received much attention as other diseases. So they are called as neglected tropical diseases. So neglected tropical diseases or NTDs mainly affect the poorest countries in the developing world. That is correct. Dengue, leishmaniasis, filariasis are few examples of NTDs. That is also correct. All NTDs are transmitted through vectors like mosquitoes, black flies, etc. No. NTDs are common in those areas where people do not have clean water and proper hygiene. That is correct. So 1, 2 and 4 is the right answer and it is option C. Then next one. Bowman-Brick inhibitor is a protein molecule with a defensive role and inhibit the activity of proteases such as trypsin and chymotrypsin. A novel Bowman-Brick inhibitor is purified from seeds of Rhynchosia sublobata, a wild species of cultivated pigeon pea, and it is named as RSBBI. The RSBBI in its pure form is tested for its stability against temperature, pH and DTT. The stability of RSBBI is expressed in terms of % control of its residual trypsin inhibitory activity and chymotrypsin inhibitory activity. So there is a inhibitor molecule, that is Bowman-Brick inhibitor molecule, which has defensive role and inhibit the activity of proteases, that is trypsin and chymotrypsin. Now they have isolated a new BBI from Rhynchosia sublobata and named it as RSBBI. Now RSBBI is in its pure form, that is they purified it and tested for its stability against temperature, pH and DTT. And stability of RSBBI is expressed in terms of % control of its residual trypsin inhibitory activity and chymotrypsin inhibitory activity. Now the following conclusions are drawn from the figure shown above. So this is the figure, I have pasted it again here. The CI, that is chymotrypsin inhibitory activity of RSBBI is stable from 37 degrees to 100 degrees Celsius, while TI activity of RSBBI is stable from 37 degrees control to 90 degrees Celsius and lost less than 10% of its activity at 100 degrees Celsius when compared with the control. That is correct. Why? Because if you see this graph, you can see that at 37 degrees Celsius, CI activity is stable. While TI activity, this is the black shades, the TI activity is stable from 37 degree to 90 degree only, that is over here. And at 37 degree control to 90 degrees Celsius and lost less than 10%, but when it reached 100%, it lost 10% of its activity at 100 degrees Celsius when compared with the control. So first statement is correct. Then next statement, the CI activity of RSBBI is stable from 37 degrees Celsius control to 100 degrees Celsius, while TI activity of RSBBI is stable from 37 degrees Celsius control to 90 degrees Celsius and lost 90% of its activity at 100 degrees Celsius when compared with the control. It is not 90% of its activity is lost, only 10% of its activity is lost. So option 2 is not correct. Then exposure of RSBBI to 1 millimolar DTT resulted in more than 90% loss of its TI activity. DTT, this is the graph. So this is the TI activity, that is shaded ones. So it resulted in more than 90% loss in its TI activity. At 1 molar, yes, over here there was 90% loss of TI activity and approximately 50% loss in CI activity. This is the CI activity, so there is 50% loss in CI activity over here when compared with their respective controls. So these are the control values over here, 100%. So there is loss in 90% and for TI activity and 50% in case of CI activity. So 3 is also correct. Then next one. The TI activity of RS-BBI is stable between pH 5 and 7. No, it is not stable between 5 and 7. Over here there is a decrease in TI activity. While CI activity of RS-BBI is lost by approximately 50% between pH 5 and 7. That is, no, there is not much loss. So option 4 is not correct. So the answer here is 1 is correct and 3 is also correct. So 1 and 3, option A is correct. Then next one, identify the correct matching. Pox virus, aggression, herpes virus, two different diseases by same virus, adenovirus, intracellular enveloped virions. They are non-enveloped. So incorrect matching is option C. Then next one, which of the following statements are false? T and B cells are produced in bone marrow. Yes. T and B cells are matured in the thymus. No. T cells mature in the thymus and B cells in the bone marrow. So option B itself is false. T and B cells play key role in cell-mediated immunity. Yes, T and B cells have memory. That also is correct. Which of the following statement is true? Immunoglobulin A has the subclasses. Yes, it has IgA1 and IgA2 subclasses. So that itself is true. Immunoglobulin M exists in monomeric form. It is in pentameric form. Immunoglobulin D crosses the placenta. Not D, G crosses the placenta. Immunoglobulin G acts as a receptor for developing B cell. It is immunoglobulin M that acts as receptor on developing B cells. So only option A is correct. The next one. The correct order of formation of the membrane attack complex formation in the classical pathway of the complement system is. So it is option B, that is binding of complement component C1 to antigen antibody complex, splitting of C4 and C2 components to form C4b2A, splitting of C3, formation of C5 convertase. Then next one. Which of the following is a part of gene ontology description? So it is molecular function. Answer is option A. Then which of the following are primary databases? So GenBank is a primary database. DDBJ is also a primary database. The other two are not. So 1 and 2 are correct. Answer is option A. Then Clustal Omega. Clustal Omega is multiple sequence alignment tool. BLAST is a sequence search tool. Blossom is a substitution matrix and GenBank is a comprehensive database of nucleotide sequence. That was very easy. Match the following. Answer is option B. Then to visualize a protein molecule structure, which of the following tool can be used? So the tool that is used for visualizing protein molecule structure is RASMOL. Answer is option B. Then which of the following methods is not associated with prediction of protein-protein interaction? So it is Newick method that is not involved. All the other methods are involved in prediction of protein-protein interactions. Then GOR method is known to predict. So it is known to predict secondary structure of a protein. It uses both information theory and Bayesian statistics for predicting secondary structure of a protein. Then next one. Which of the following is not a force field? So it is GROMAX. Answer is option A. Then arrange the following steps involved to perform homology modeling in the correct order. So first will be selection of the template. That is 1. Then comes alignment of the template and target sequence. 2. then comes backbone model building 3, then comes model refinement, and then model evaluation. So it will be 1, 5, 3, 4 and 2. So that is option D. Then protein engineering is the process of developing useful or valuable proteins. Find the correct order for the basic process of protein engineering. So first one will be prediction of protein function 3. Then comes design of protein structure. Then comes nucleotide sequence derived from amino acid sequence, and then DNA synthesis. So it is 3, 1, 4 and 2. Option D is correct. Identify the correct pair of chromosomal disorders. Down syndrome. Which pairs are correctly matched? Down syndrome, it is 2, that is trisomy 21. Okay, your B itself is correct because B is the only one having 1, 2. Okay, we'll see the others also. Klinefelter syndrome is extra X chromosome. Fragile X syndrome is FMR1 gene on the X chromosome, and Turner syndrome is missing X chromosome. So the answer here is option B. So coming to the next question. The expression of the gene can't be determined by. So expression of the genes means the protein is produced. So it cannot be determined by southern blot hybridization. So the answer here is option A. Whereas reporter gene analysis, western blot analysis, immunofluorescence assay give result on proteins. That is expression of your gene. But southern blot hybridization, it doesn't give. It is based on DNA analysis. The next one, bacterial genomes are protected from its own endonucleases by. So they are protected by methylation at restriction sites. Answer is option B. Then match the correct labels for the brain parts. So first one is frontal lobe, second one is parietal lobe, third one is occipital lobe, fourth one is temporal lobe, and fifth one is cerebellum. So the answer is option A. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.